In the first lecture of this series about the introduction to molecular genetics, we talked a little bit about the history, the early history of DNA back in the 1800s when they had actually discovered this item they called nuclein because they thought they came from the nucleus. And then later on, the fact that the nuclein was divided up and they understood that there were 4 primary bases that were part of nuclein, and they're called nucleic acids, as well as uracil, the 5th. But then again, there was controversy because chromatin had been discovered, and chromatin was known to be a combination of these nucleic acids plus proteins. And people said, well, there's no way that this just simple 4 bases can code for all of life. That's not possible. And it must be carried in the protein. So even though there's somehow all together there's nucleic acids and proteins mixed, it's probably the proteins that are carrying the information. So by 1920s, this was a sort of an ongoing battle and an important question. And so some sets of experiments started that led us almost a half century later to finally understand and finally accept that DNA was the genetic material. So I want to address the first of these 2 experiments. And these 2 experiments, as we said last time, are working under this model of what's known as the transforming property. The idea that one of these organisms, the material from one organism, could affect another organism or transform it. Okay. Now, where this came from was a series of experiments that were published by an individual named Griffith in 1927. Griffith was working with the health ministry in the UK in London. During the Second World War, one of the major killers for anybody, okay, besides tuberculosis, of course, was still there. Remember, now this is a period of time when we had yet to discover very effective antibiotics. Certainly things like even as simple as penicillin were yet to be discovered. So if there was any type of infection or any type of other form, really it often led to mortality. And certainly one of the big ones from that was pneumonia, okay? And in fact, for a lot of the people that were in World War I, in the trenches, and it's cold and wet and things, pneumonia was a big problem, as it was in other conditions. Well, Griffith was working to try to understand about pneumonia. And one of the things that had happened, of course, by this period is that sterilization had become a common medical technique. Now, believe it or not, back in the late 1800s, people didn't sterilize anything. Things were, the same knife was maybe wiped off and used for 4 experiments, or the same needle was used for 4 or 5 different operations or whatever, and with very little concern for sterilization. But because of Pasteur discoveries and others, people understood that now that you could sterilize things by heat and pressure. Okay. So what did we start out with? Well, we had this idea of pneumonia. We had some idea that pneumonia was caused by bacterial infection, and that there were really 2 forms of this bacterial infection. There was what was a virulent form. Okay, this was known as type 3S, S standing for smooth bacteria. And then there was an avirulent form of this bacteria, a non-virulent, and it was 2R, rough. So rough-coated ones did not normally produce any type of major infection. They could be resisted. The smooth forms were the virulent forms. All right, so what did Griffith do? Well, what Griffith did was a simple experiment, okay? He said, okay, I'm going to set up, as you would, any type of controls. The controls being, I'm going to inject the non-virulent form into a mouse, a living organism, happened to be a mouse, and see what happens. I'm then going to inject the virulent one, the one that would cause the disease, and make sure that my model works, that in fact the mice respond to it by having essentially pneumonia, by having a bacterial infection. And then I'm going to go through this process of sterilization. I'm going to heat kill or sterilize the bacteria, this virulent form, and inject it into the mouse. And what I expect to see from that is that the mouse will not die. Okay, they not become infected because I've effectively killed a disease by sterilization. And then, very amazingly, he decided, you know what I should do is I should take a mixture of this rough form, this non-virulent form, this living, okay, and take some of the heat killed over here and just mix them together. Let them put them together and see what happens when I mix them together. So I've got a living form, this rough form that's totally harmless, okay, I've got a heat kill form that by itself cannot kill the organism, and I'm going to mix those two together. This is truly a sort of a Frankenstein experiment, as I said before in the last lecture. So what were the results? Well, the results were, from the controls, exactly what you'd expect. You got the virulent form kills the mouse, the non-virulent form injected, the mouse lives. It's these other two that are very interesting for us. So let's look at those, sort of pull them out into detail here, okay? So we've got this virulent material, the material that would kill you. You inject it into the mouse. As predicted, the mouse gets sick and dies, okay? You take the secondary control, the avirulent material, you inject it into a mouse, and it lives. So the avirulent material is living, okay, so living bacteria. You inject it, the mouse is able, its immune system is able to take care of it. Okay, then you start the real guts of the experiment. You heat kill, you sterilize the virulent material, inject it into the mouse, the sterilization technique worked, the mouse lives. You've eliminated the ability of this virulent form to actually to replicate and cause death of the organism. Okay, and then what happens when you do the other experiment? You take the avirulent form, okay, so that's the living material that couldn't hurt you. You take the heat-killed virulence, which is part of it that can't hurt you now because it's heat-killed. You mix them together, give them a little time, you inject them into the mouse, and lo and behold, what you see is the mouse dies. Okay? It's gone back. Somehow, the information that was in the heat kill that's bad was incorporated into this avirulent, this harmless living material in some way. Okay? So the conclusion is that something in the preparation transformed or changed, in this case, an avirulent material into a virulent material. There was a movement of information from stuff that was dead, essentially, into something that was living over here. There was a transformation. Okay? Well, as you can expect, there was a lot of objection, a lot of concern about this experiment, a lot of doubt that it even was correct. How could this be? How could you take a dead something and it take over a living something? And it went on for a number of years with a lot of concern. People tried to replicate it. Sometimes it worked. Most of the times it worked, and sometimes it didn't, and there was a lot of back and forth about it. Okay? Well, in 1944, there was a group of scientists who were well aware of Griffith's work, and they had some new technology. And very often in genetics, new technology is what drives the system, right? That's what provides us with new things. And in this case, there was new technology, and they could go back and they could repeat the experiments of Griffith, and they could more precisely isolate what among the possible items really was the transforming property. Okay, so they were interested in what was the transforming agent. What really was it? Okay, they decided, as being scientists, that they would, of course, apply the scientific method. Now, what does the scientific method say? Scientific method says, all right, what we're going to do is we're going to propose some ideas, okay, hypothesis. We're going to propose some predictions from that hypothesis, and we're going to try to see which one of those predictions are wrong. And ultimately, once we've eliminated everything that can't be correct, okay, we've eliminated things that are wrong, what's left, hopefully, is going to be the final thing that's correct, right, the last thing. That's the way science works. Science doesn't prove anything. We talk about that later in the lab. One of the big points we make is science doesn't prove anything. What the scientific method is designed to do is eliminate those things that can't be possible with the idea that whatever's left over may be the truth, may be system. Okay, that's how science works. Well, that's exactly what they did. They said, by the 1944s, we have a pretty good idea that we're dealing with things like RNA, DNA, and proteins, right? The makeups of chromatin, as well as fats, lipids. Okay? What's very important, and what the technology change that occurred at that time is that there had been isolated enzymes that specifically targeted each of these groups. RNase had been isolated and purified, so RNase destroys RNA, selectively destroys RNA. Proteinase had been isolated, and proteinase selectively destroys proteins, right? So you can get rid of the proteins without killing the RNA. Then also by that time, DNase had been discovered and isolated, and they had access to it. DNA selectively destroys DNA without going after RNA or without going after proteins. Also, there were lipases known. Lipases are enzymes that destroy lipids and break apart lipids. Okay, so now they've got the tools. They can selectively go in and they can eliminate one or the other. And that's exactly what they did in their experiment. Okay, what they did was they heat-killed cells. They took those cells and they treated them with one of these combinations. Okay, basically repeating the experiments of Griffith, but by this time, by an extra step, they would go in and target one of the elements. So they would take RNase and they would add it to these heat-treated cells. They would take those heat-treated cells, put them into a culture in a similar way that Griffith did, not exactly, but similar. They would put them, for our purposes, they would mix these RNase-treated, avirulent, heat-killed with live cells. Okay, that's not exactly what it did, but that's close enough. All right. Now, they would then take that and they would look to see if there was transformation. Sure enough, there was. In other words, what that's saying is, if you go through the same experiment as Griffith's and you remove the RNA, you still get transformation. Okay? They then said, okay, well, let's try it with something else. Let's try it with proteins. Let's kill all the proteins out and see if we get transformation. So they took proteinase and they killed off the proteins and they said, okay, do we get transformation or not? Sure enough, they got transformation. Okay? So what have we learned so far by the sort of the steps of science? We have predicted that RNase could be the genetic material, or RNA could be the genetic material. We've eliminated RNA, and you still get transformation. So that's not what caused the transformation. We've proposed that protein was the genetic material, right? That's what makes up that's the likely thing that everybody thought it was. And sure enough, you get rid of the proteins, you still get transformation. All right, well, now we got to go and we got to set the other parts up. They did the same thing with lipases. I didn't show it here, but same thing, okay? All right, when they do that, what you see is this, okay? Let's take the other element. Let's take DNA and let's get rid of it. Let's use DNA and eliminate DNA. Do the same basic experiment, but selectively eliminate DNA, and lo and behold, no transformation. All right? Absolutely no transformation occurs. So if you take out DNA, stuff starts to happen, okay? This experiment supports the idea that DNA is capable of transformation. Supports it, okay? I didn't say it proves it, okay? In fact, a lot of people didn't think it did prove it. They thought that, well, what really is going on is something else, okay? And we'll talk about that in a moment. But it certainly supports the idea that DNA is capable of transformation. So let's go back and put this in context of Griffith's experiment, just like Avery McLeod and McClarty did. Okay, so they take the proteinases, they go through the experiment of mixing all this together, which in Griffith's experiment would kill the mouse, and lo and behold, it still dies, right? You remove the RNases, so you remove the proteins, it still dies. You remove the RNases, it still dies. So the transformation has still occurred. Okay, finally, lipases still dies. The only thing that stops this process is removal of DNA. Once we do that, the agent is destroyed. In other words, we have stopped the process. Okay? So their conclusion was that the chemistry of transformation lies in a molecule whose composition is DNA, is nucleic acids. Right? Beautiful set of experiments, well done. Finally put the nail in the coffin. Okay, whatever's doing all this transformation, it directly involves DNA. Okay? So that was where we left. And this is in the early 1940s, okay, the middle of 1940s, and this is the state of the science. So this is where we'll stop this lecture. In the next lecture, we're going to come and we're going to take this one step farther because we haven't really convinced everyone in a definitive way that DNA is the material. We'll explain why that's true in the next third edition of this particular series of lectures.